Welcome to the another session of the series of talks marking the 200th birth anniversary of the father of genetics, Gregor Mendel. I am Visha Gupta, pursuing PhD in Dr. Sridhar Sivasubbu's lab at CSR IGIB, New Delhi. With immense pleasure, I would like to introduce today's speaker, Professor Pradeep Kumar Verma. Currently, he is a professor at Department of Genetics of University of Delhi and Vice President of Indian Society of Cell Biology. Also, he serves as a member and chairperson of several academic and administrative committees of University of Delhi. In the past, he has served as the Dean of the Faculty of Interdisciplinary and Applied Sciences and as the head of the Department of Genetics and Department of Biophysics. He is an excellent teacher and has been a part of several programs which aim towards teaching science to young minds. He has been awarded the INSA Teachers Award for the year 2016 by Indian National Science Academy, New Delhi. Also, recently in 2021, he has published a research article titled An Alternative Approach to How Biology Practicals Are Currently Conducted in the Indian Education System in the GPC Journal of Science Education. So broadly, his research area includes studying the expression of transgenes in plants and development of transgenics for hybrid seed production in crops like cotton, tobacco, brinjal, etc. Welcome, sir. Thank you for accepting the invite for the session. So today we have some questions on that, which we like to have your thoughts and views. So firstly, sir, can you brief us about the research that is currently being done in your lab?
Well, thank you, Vishu, for that elaborate introduction. Of course, the kind of work that is happening in our lab is to do with plant biology. There have been two aspects of it. One is to what one can talk about in terms of basic questions and also to make products. In the area of asking basic questions, my main interest has been on promoters, basically, of course, plant promoters, some of which have been to develop synthetic constitutive promoters, analyze some of the constitutive promoters, and more recently, my interest has been to look into tissue-specific promoters, for example, the tapetum-specific promoters like TA29 and A9, and figure out what are the different kinds of transcription factors and cis elements that make such promoters tissue-specific. Overall, apart from looking at promoters, my interest also lies in how to improve or how to understand improve transgene expression in plants. And that stems from the fact that we have been involved in developing different kinds of transgenics. And the two major transgenics that I have worked on in collaboration with Professor Paintel has been in developing the Barnase bar star based pollination control mechanism for hybrid seed production in mustard. And the second has been in developing insect-resistant cotton using the very well-known BT gene or the Cry1AC gene. So this in brief has been the kind of work that happens in our lab, both some part of it which has been sort of the development of transgenics, which has potential in the farmer's field. And of course, the other part is to keep answering some of the basic questions.
Okay, sir. So yes, the research that on the development of transgenics is, I think, the direct application of what Mendel's discoveries have shown. So this is like a very advanced version of that. So how do you think that Mendel's discoveries have impacted your research and the research that is being done 150 years down the lane of those discoveries?
Vishu, that basically all this work, if you really look at anything which has to do with manipulating of genes, or be it through a transgenic approach, or be it through basic classical approaches which are non-transgenic in nature, actually has a foundation in what Mendel has observed. If you really look at it, his observations, of course, at that point of time, he had not looked at it in terms of, he talked about it in terms of factors, but came out and laid down some of the basic principles by which these factors get inherited from generation to generation, and brought about some of the very basic concepts. So in that sense, if you really look at it, most of the work that is done in the area of genetics will have a foundation back in the way that in what Mendel had talked about in his seminal paper way back. And of course, if you really look at it in terms of direct application, if you want to talk about if there's a direct link, because you all familiar with Mendel talked about inheritance of dominance, recessiveness, segregation, independent assortment, and those actually even find when you develop transgenics, you have directly, like when you are making a transgenic, you talk about that this is going to be a dominant, it will have a dominant effect or show up as a dominant phenotype. We study segregation when we are looking at this inheritance of the genes, transgenes, and looking and using these values that we get in a segregating population. We can answer questions like whether there's a single integration of the transgene in a single locus or at multiple locus. So actually nothing which you do where you are manipulating the genetic elements and so on, genomes and so on, you will have to rely on some of the things which Mendel talked about.
Yes, sir. So truly, the Mendel's discovery were very significant in what we do research today. This leads me to the next question: that why do you think it is important to study genetics? Means what is the application of studying genetics and then taking it to the research?
See, this just like what I mentioned when you're talking about genetics, and in terms of genetics, it just does not. Mendel started it all off in a way. If you really want, if I were to put it that way, he started it all off. He asked very pertinent questions. Even if you look at before Mendel, it was not that Mendel was the first one who did hybridizations. Even if you read his actual original paper, he talks about others like Bushera, Nageli, and others who were doing plant hybridizations. But he was the one who really asked those critical questions and tried to figure out what happens when you hybridize two different varieties of plants and how is these phenotypes inherited in future generations, and I'm sure everybody is taught about it, which lays the foundation. Of course, that started the whole story, which again had to wait for a long period of time before it was rediscovered. And then, of course, others started to ask these questions, like one was looking at it as factors. And then this led to a question where like people like Morgan and others started becoming interested in where is this abstract thing lying? Where are these factors? Is it in the cytoplasm? Is it in the nucleus? And that led to the chromosomal basis of inheritance. So then people started talking about that, okay, these factors are things on the chromosome, which led to the DNA, what is made of the genetic material, or what makes up a chromosome in that sense. So it led to the DNA and so on, and different kinds of questions came about. So if you really look at, but even before you had the structural entities, knowledge of structural entities, there was so much of work which talked about how these factors interact or how do these different kinds of phenotypes arise. To the extent you had talks about gene interactions, how you can have different kinds of new phenotypes coming out because of different kinds of genes or genes interacting and so on. And so if you really look at the history, it started off with Mendel, and then you had all this work on poultries like chickens and other things, and then of course moved into fly rooms and so on and so forth. So see, the basis of all this lies in studying of genetics. So as a student today, one can ask, what is the use of studying those old paper of Mendel of talking, not really old, it's probably not the right word, but like, okay, why do we need to study a Mendel? All I need to know is, okay, there is a 3:1 ratio or a 9:3:1 ratio. But I think its understanding is very, very important. This basic concepts understanding is very important to bring on. If I understand independent assortment, then it makes sense for me to understand linkage. And from linkage, we have genetic maps. Today, when you talk about I'll give an example from the crop side. We have all this work on crop breeding, where what you want to do is you want to introgress characters of importance from one variety into a variety which you are going to use. And for that, you need genetic maps. You want to get into identify the genes which are involved, which gives you some interesting characters or phenotypes. So for that, you need a complete understanding of how today you may be working. Initially, if you looked at geneticists working, they would work with 1 or 2 or 3 variables at a time. That's what I'm talking about, are variants of phenotype. today with DNA as markers for the variations available in a given genome, I can work with hundreds of them together. But they will still follow the way these DNA markers will work, will still follow the same kind of observations that geneticists had, be it segregation, be it independent assortment, be it linkage. So that's why I feel that studying genetics or studying all these concepts is very important, even in the day-to-day where you may be working more with DNA markers and fewer phenotypes, but I think that lays down the foundation of the whole system.
Yes, sir. So
I hope that answers your question, Vishu.
Yes, sir. Yes, sir. Definitely. So, sir, it is very like as we have seen the advancements that are coming up in the field of genomics also. So, if we do not have that basic knowledge or the like why we what is the basic principles of genetics, I don't think that those advancements or those things would be helping us to study the actual gene structures or actual DNA elements that we study using those genomic approaches. So, sir, that is why I would like to now have your views on what do you think that the current advancements that have been taken place in the field of genetics and genomics, like how it will be helping to improve the genetics in the future, the current advancements that are taking place in the field?
See, one of the things that has happened today is, just like what I mentioned, if you look at it, I'll give you an example to make this, how has these advancements helped? And I'll again take an example from crop breeding, for example. So for example, I'm sure you would have heard about that introgressions of a trait from one, let's say from a wild variety into a cultivated variety. So let's say, for example, I have a disease resistance in a wild variety, a resistance to us, let's say a particular some disease, which I now want to introgress it into the cultivated variety, so that the cultivated variety also becomes disease resistant. So earlier days, and this can be done by what is called as backcross breeding. So basically what you do is you make a cross between the resistant variety and the wild and the sensitive variety. And then you introduce this gene of interest, which you are now following just through a phenotype, that whether it is resistant or not. And then you keep backcrossing it with recurrently with the parent, which is the cultivated variety, so that you finally you just have the gene of interest from the wild, and you reconstitute the whole of the genome of the cultivated, so that you can grow it in the field, get the advantage of the cultivated variety, which now also had as an additional feature of being resistant to a disease. Now, when you didn't, in the earlier times when breeders used to work on it, you had phenotype only to track this variation. So you would have to, the disease resistance variation could be tracked very easily because you had a way of testing whether a progeny is resistant or not. But tracking the background, that is where we are replacing it to the wild type variety, was difficult. It depended largely on breeders' eyes, that, okay, which of the plants look more like the cultivated variety in the progeny and so on. The breeders' eyes would try to pick up the best lines which would be, which they could take forward in the breeding program. Now look at it with the advent of knowledge of DNA markers, where you are directly not bothered about doing, you have the phenotype, but you can directly look at the variations between the two parents through DNA markers. Now you can do much more precision breeding. That is, I don't have to depend solely on my breeder's eye to look at the phenotypes of what is my backcross population looking like. Using markers, I can identify which one is more closely constituted to the backcrossed parent. So that is, these kind of technologies have made more precisions in the way these earlier breeding was happening way before we even knew DNA sequences and genome sequence and so on. All that it has happened now is that we have now got tools to do it in a more precise way. So let's say, for example, if I have a resistant character, to be able to see it phenotypically, unless I make it homozygous, I will not be able to see that resistant character. However, now if I have a DNA marker 100% linked to that resistant gene, then even in a heterozygous situation, I can find out whether my resistant allele, which is a recessive allele, is, let's say, a recessive allele is present or not. So those kind of things have happened through. Of course, The other thing that it has opened up is earlier people would only do forward genetics. That is, that I had a mutation, I had a phenotype, I had a variant phenotype, and we would then try to track what the gene is, what is the gene that is responsible, or the variation that is responsible for this phenotype. Today, you have genomes available to you. And now what you are doing in many of the cases is what we call as reverse genetics. You have an unknown piece of sequence, and then you try to figure out that what does this sequence do. So, the way that you answer questions today, or the way that the kind of questions that you can ask today has improved with our knowledge and the various kind of tools that can be applied in today's terms.
Yes, sir. So talking about the Mendel, from the Mendel till today, we have like so much advancement that has improved our understanding of the genetics, and also helped us to take steps involving genetics to solve many of the problems that are public, or in case of human genetics, if we look at public health problems, and in case of plant genetics also, the crop improvement programs. So I hope this session would be helpful for all the young minds to bring the curiosity among them to learn and study genetics. So thank you so much, sir, for sharing your enlightening views about the field of genetics. Thank you, sir.